Biology chapter 18 theory video 12. Now we learned how to use Simpson's index of diversity. So the advantage of using Simpson's index of diversity is that we don't have to identify all or even any of the species that are that are there in that sample. We can just simply call them. Now, for example, if there is a species of anemone with short tentacles, we can call it anemone A, and if there are few long tentacles, then anemone B. So we say that we can identify them to be different species, but again we have to be very careful because there could be same species with different phenotypes, so we have to be careful as well. So then when we get a higher number for D, we know that it has more biodiversity. If it is less, then if the value of D is less, then we know that it has low biodiversity as well. Then also with regards to this biodiversity, we have to understand that if there are 10 species in two areas, in one, only one type of species is most abundant. All the others are in very few numbers. In the other one, there are few species who are greatly in abundance. Then the second situation where there are many species with relatively high numbers of organisms will have uh, more biodiversity than the first situation where there was only one type of species having high numbers of organisms. So all of these things need to be taken into consideration when discussing biodiversity. Now we are going to move into systematic sampling. So random sampling is not suitable for all the situations. Sometimes when there are physical conditions changing, and such as altitude, soil moisture content, pH, or exposure to light intensity, etc., if we are considering these situations or changes in these situations, we need to we might want to take different or selective sampling. So we might have to take not random but selected sampling. So that means we are going for systematic sampling. Um, there are two methods that we can take systematic samples. First one is taking a transect, that is to recording the drawing a line across across a range of physical features, and then identifying which plants or which organisms that touch that line at certain distances, for example every 2 meters. So that will give you this line will give us the quantitative data that we can actually present using graphs and drawings, etc. So this phenomenon is actually used when there is a physical feature that changes, and when there is a requirement for us to select certain areas or habitats. We have to if we have to select them rather than going for the random observations. So then also we can actually use we can place quadrats at regular intervals along this line and record the different abundances of each species within that quadrat, and also we can observe or take those data, and then we can plot them in a bar chart or a kite diagram that will be coming in worked example 3. That will also help us to understand how they work. So the keyword transect is a line marked by a tape measure along which samples are taken either by noting the species at equal distances that is line transect, or placing quadrats at regular intervals, that is belt transect. So this is line transect. As you can see, it is along the line, then the organisms found at regular points along that line are noted. So if we are going to detect changes in community composition and their frequency and density, then we can detect them. So the main important thing is actually it could be the possibility of analyzing one or more habitats is quite important in this particular event. Then interrupted belt transect, as you can see, the abundance of organisms within quadrats placed at regular points along a line is noted. So as you can see here, we have a single quadrat, then here another one. So they are placed at regular intervals, but they are not random, as you can see. So then we compare those situations also, and we can figure out how they are related to one another. Let's write down a note before going on to the question. Write down. When using Simpson's index of diversity, we do not need to identify all or any of the organisms present to the level of species if we are able to recognize them as different species. We can name them differently and find their diversity. Leave a line and write, biodiversity not only depend on the number of species, but also their abundance, Next topic is systematic sampling. When we are required to investigate how species are distributed in an area where the physical conditions change, within brackets, altitude, soil moisture content, soil type, soil pH, exposure, that is light intensity, etc. Close brackets. We use random sampling, we use systematic sampling rather than random sampling. Leave a line and write, there are two methods. First is sampling the organisms along a straight line at set distances . This is known as a transect. Put a dash and write down line transect. Next, placing quadrats at regular intervals along the transect and comparing their abundances. Put a dash and write down belt transect. Now, so this is line transect and belt transect. This belt transect is also known as interrupted belt transect. So you would be able to understand it better. Then we move on to question number 3. some students used an interrupted bed transect to investigate the distribution and abundance of seaweeds and mollusks on a rocky shore, placing a quadrant every 20 meters. The students began sampling at 20 meters below mean sea level when the tide was out and worked their way up to the shore. They used two different scales for assessing the abundance of seaweeds and mollusks. On the scales, 5 indicates the highest level of abundance and 1 the lowest. If a species is not present in a quadrant, it is obviously recorded as 0. Table 18.9 shows the data collected from an interrupted bed transect on a rocky shore. The data are presented in the kite diagram in figure 18.29. So this is the details that they have given. So as you can see, you will have to study this, with long exposure. You will have to look at this very carefully. This is not going to be something new, but this is going to be a different way of presenting the same old things, right? So this will give us the kite diagram as to their relative abundances. So I hope you understand. If you have any doubts, please let me know. And so as you can see, that they are presented in according to the distance from the shore and their relative abundances. If you have any doubts, please let me know. I will explain it to you further.